Okay, so welcome back from the break. Whether you stopped and went and got a drink or whatever or you just clicked and continued on. Let's take a look at those concept check questions, and then let's take a look at DNA replication, which is going to be the subject of this section. So if you've got this polynucleotide sequence, how can you identify which is the 5' end and the 3' end? Well, as it stands, you can't, because you're not told which has the phosphate group, which would identify the 5' end, and which has the hydroxyl group, which would identify the 3' end. So the information that you're going to need is which of the nucleotides, the G or the C, has the phosphate group that would tag the 5' end, and which of them has the hydroxyl group, which would tag the 3' end. So that's the information that you're going to need. So we could put in here we need to know which nucleotide has the phosphate group. That gives you the 5' end. So Griffith was very surprised when the mouse injected with the dead S cells and the live R cells died. What result do you think he might have expected, and why? Well, if you remember, I kind of gave you the answer to this question. We know the dead S cells don't kill the mouse. We know the live R cells don't kill the mouse. So if you're injecting two types of cells that don't kill the mouse, the expectation would be the mouse is not going to die, because neither of these cell types are pathogenic. Neither of these cell types are deadly under the circumstances. So it is very, very certain that Griffith was expecting the mouse to survive. So to wind up with a dead mouse was something that was totally unexpected. And the why is, of course, because neither of these cell types are pathogenic. They shouldn't have killed the mouse. It should not have died, but it did. And it was the presence of the living S cells that told him why the mouse died. But it was trying to figure out where those living S cells came from that was the huge insight that Griffith had as part of the explanation of his experiment. So those are the answers to these two concept check questions. So let's talk about DNA replication. So remember, Watson and Crick had implied kind of in their original paper that it was semi-conservative, and had been a little bit more explicit in implying that it was semi-conservative in the second paper that they co-wrote with Wilkins, that was published in that series of 3 papers about DNA structure in the April 25 edition of Nature in 1953. So what does semi-conservative replication actually mean? Well, here is a simplified explanation of what happens. You have your original DNA molecule with the A's paired with the T's and the G's paired with the C's across the DNA molecule, cross-linking. So we say that A pairs with T, or that A is complementary So the 2 strands are considered to be complementary strands. So while we often refer to the base pairing rules as A pairs with T and G pairs with C, and we can often say that, and you'll often see that said, the proper terminology is to say that A is complementary to T and G is complementary to C, and the two strands are complementary to each other. And so you're going to see a lot of reference to complementary strands when we talk about DNA replication. So we start with our DNA, our double-stranded DNA molecule, the parental molecule. We unzip the strands. And in fact, that's the technical term that is used. We actually say that they are unzipped because it's kind of like undoing a zipper. And then the two parental strands act as templates. And because they represent half, they each represent half of the original DNA molecule, that's where the concept of semi-conservative comes from. Half, semi, of the original DNA molecule is always conservative, in each of the new DNA molecules. Hence the name semi-conservative replication. So we take our original parental DNA molecule, we unzip the two complementary strands, and we use each of them as a template to generate the missing complementary strand. When the two missing complementary strands are completely regenerated on the templates, we have two new DNA molecules where once there was a single DNA molecule and replication has taken place. The problem for the two researchers who published the paper that definitively showed that DNA is replicated in a semi-conservative fashion. Two individuals named Messelson and Stahl, again, associated with the NIH laboratories in Washington, DC, was to differentiate between the original parental DNA molecules and any daughter DNA molecules that might be generated as a consequence of replication. Because not only were they trying to show that DNA was replicated in a semi-conservative fashion, they had to be able to demonstrate that it was not replicated in some other fashion, and two alternative models had also been suggested. One is known as conservative and the other is known as dispersive. The problem for Messelson and Stahl was, how do you separate out the original parental molecules from any new DNA that was generated? So what do these three models propose? Well, we already know what semi-conservative looks like. You start with your original parental DNA molecule. You unzip the original DNA molecule. Each of the original, one strand of the original DNA, or each strand of the original DNA molecule then serves as a template to create new DNA molecules. When you do a second round of division, the original DNA strands are still there and they are used again as templates, but now the new DNA from the first round of division is also going to be used as templates. And so you're going to have more new DNA, quote unquote, new DNA being produced. And over time, there'll be an increase in the amount of new DNA, but you're still going to have a small amount of the original DNA molecules present all the time because it's being conserved. The conservative model said that when new DNA is formed, the whole of the original DNA molecule is conserved, fully conserved in its original form, but it somehow serves as a template to make DNA molecules that are completely new on the basis of the original DNA molecule. Now, what we have to remember here is that Messelson and Stahl are not trying to figure out the actual mechanics of DNA replication. All they want to know is it semi-conservative or is it not? Because Watson and Crick had predicted it would be semi-conservative. So they're not trying to explain how it's done, just whether it's done in a semi-conservative fashion or not. The third model is known as the dispersive model, where there is no attempt to maintain the original DNA molecule intact or even one of the strands of the original DNA molecule intact. But as DNA replication occurs, you get a sequential dilution of the original DNA by the insertion of new DNA as a consequence of DNA replication into the strand. So over time, you get a sequential dilution of the original parental DNA by the production of new DNA. So Messelson and Stahl were aware of what these three models were implying. The problem was, how do you separate out the parental DNA from the daughter DNA molecules? Well, it turns out that the DNA molecule itself would provide the solution, and it was in the form of those nitrogenous bases. They contain nitrogen, and it turns out that nitrogen atoms exist as two isotopes. Nitrogen 14, which is by far the most common form of nitrogen, but a slightly heavier isotope also exists called nitrogen 15. The difference in mass is minuscule. It's a single neutron in the nucleus is the difference. But the reason why this was significant was that if you go back and take a look at the nitrogenous bases, you'll understand why they're called nitrogenous bases, because they have multiple nitrogen atoms in them. And if you look at the average DNA molecule, and in the case of Messelson and Stahl, they were going to work with E. coli DNA because growing bacteria is really easy. The E. coli DNA contains several 100,000 base pairs. Now, a base pair is two nucleotides, so they contain many hundreds of thousands of nucleotides, each containing at least two or more nitrogen atoms. If you could replace all of those nitrogen atoms in a typical E. coli DNA molecule with nitrogen 15 instead of nitrogen 14, you would create a type of DNA molecule that would be substantially more massive than a DNA molecule that contained only nitrogen 14. And it would be massive enough that you could separate it out by a technique called differential gradient centrifugation. Basically, you create inside a centrifuge tube, and I'll draw a diagram here for you, a continuous gradient a solution where the density of the solution increased as you move down the centrifuge tube in a continuous fashion. And the reason why this was their chosen technique was simply because DNA molecules placed in the solution will get pulled down through the solution by the artificial gravity field as the centrifuge is spinning, and will simply move through until they reach a density that is equivalent to their mass. So if you've got two DNA molecules, one that contains just nitrogen 14 and one that contains just nitrogen 15, they're going to move to different positions in the centrifuge tube. So you might get a band of DNA high up in the tube that contains the nitrogen-14 DNA. Further down in the tube, you would get the nitrogen-15 DNA. And then somewhere in the middle there, you would get a band that contained N14 and 15 DNA. In other words, the DNA that was the product of DNA replication, where one of the strands was from the original parental DNA and the other strand was the product of DNA replication. And this is a technique that has been around for a while. I've used it in some of my research. It's a very useful technique. And pouring the gradients is not terribly difficult to do. That you can actually purchase devices that will do it for you automatically. You just make up the solution that represents the density at the top and the solution that represents the density at the bottom. You start the machine and it creates the gradient by mixing the two solutions together in a continuous fashion to create the gradient that extends from the bottom to the top. Most dense at the bottom, least dense at the top. It's a very, very simple thing to do. The only issue is running the centrifuge. You need to run it at extremely high speed, and you need to run it for an extended period of time, typically overnight. And so it has to be done in a vacuum, and it needs to be done at cold temperature to allow everything to work. If there's any air around, the friction will heat things up and will also interfere with the centrifugation. So it's always done in a refrigerated, evacuated centrifuge machine called an ultracentrifuge. And these can generate artificial gravity fields in the 100,000 G. So 1 G is the force of gravity here on Earth. These devices can generate G forces in the 100,000 plus G, would squash you to infinitely small if you could fit inside one of these things. But that's the sort of G force that you need to pull the DNA molecules down through these gradients. And so Messelson and Stahl simply took E. coli, grew them for multiple generations in the presence of nitrogen-15 so that they could be confident that their DNA contained essentially only nitrogen-15. Then they transferred that into a medium that contained nitrogen-14, allowed the E. coli to grow and divide. And of course, every time the E. coli divide, they have to copy their DNA. They then extracted DNA out of those E. coli and placed them in a set of centrifuge tubes. Now they realized that they actually needed to let the E. coli replicate twice. In other words, copy their DNA twice. Because with a single round of copying, you could easily differentiate between conservative and not conservative. Because if it was conservative, you would get two bands of DNA, one representing the heavy DNA, the nitrogen-15, and the other and the original parental DNA, and one band representing the new DNA, which would be nitrogen-14 only. If you didn't get that, well, it couldn't be conservative, but you could not differentiate semi-conservative from dispersive after one round of DNA replication, because both of them are going to give a DNA molecule with a mixture of light and heavy. You needed to let it happen, replicate twice to be able to distinguish between semi-conservative and dispersive, because in the second round of replication, remember, in dispersive, it's a sequential dilution of the heavy DNA with light DNA. So you'd still get a single band, but it would be higher up in the tube. In semi-conservative, you're going to get a band of DNA that represents half light, half heavy, because you've still got one of the original strands of DNA from the original heavy DNA parent being conserved and used as a template. But now you're going to get a second band that's higher up in the test tube that represents just light DNA. So you needed that second round of replication to distinguish between semi-conservative and dispersive. So they allowed the E. coli to grow and divide a second time in the nitrogen-14 solution, and then extracted DNA out of those E. coli and put them into the second test tube, second set of test tubes. Put them into the centrifuges and spun them up. And again, you can find the Messelson and Stahl paper on the internet as a PDF document. It was published in 1956, so just 3 years after Watson and Crick published their paper describing the double helix model. And Messelson and Stahl's results clearly indicate that it really is semi-conservative. Because what you get in the first round, after the first round of division, is this single band representing half heavy, half light. After the second round of division, you get a half heavy, half light band, plus a second band that represents light, light. It really is semi-conservative. And so in 1956, Messelson and Stahl published this paper indicating that Watson and Crick were correct, that DNA replication is semi-conservative in nature. So in 1962, enough information had been generated about the genetic code to be very confident that the information was in fact stored as the sequence of nucleotides. And of course, Messelson and Stahl had now demonstrated in 1956 that DNA replication was in fact semi-conservative. So it was considered that both of Watson and Crick's predictions based on their model had enough evidence to support them, which in turn supported Watson and Crick's model. And so Watson and Crick, along with Wilkins, were awarded the Nobel Prize in Medicine. And you notice who's missing? Franklin. Rosalind Franklin was not awarded the Nobel Prize. Now, there's a very good reason why Franklin missed out, because unfortunately she died in 1958 from ovarian cancer. And the one hard and fast rule about the Nobel Prize is that you cannot receive it posthumously. You must be alive at the time of its award, at least when the notifications go out, because the notifications typically go out early in the year, but the award ceremony is later in the year. And in fact, the Nobel Prize Committee had to adjudicate on this about 3 or 4 years ago, because an American chemist was notified that he was receiving the Nobel Prize, but in the interim between the notification and the award ceremony, he unfortunately passed away. And the committee had to meet to decide whether he would actually receive the Nobel Prize, given that he was now dead. And the decision was made that as long as you were alive at the time of the notification of the awards, then yes, you were a Nobel laureate. So he was one of the Nobel laureates in chemistry for that year. Franklin, unfortunately, was not alive in 1962. It is felt by a lot of people that if she had been alive in 1962, she likely would have been named as the third person on the award, because you can only have 3 people on any Nobel Prize. Wilkins would have actually missed out. Wilkins was given the award probably as a substitute for Franklin, because Franklin was not alive at the time. Now, for a long time, Watson and Wilkins particularly worked really hard to reduce the significance of Franklin's contribution to the discovery of the double helix structure. In more recent years, over the last 15 years or so, Franklin's role has been moved front and center, because it is very, very clear that without Franklin's image, Watson and Crick could not have achieved their model, the double helix model. It would not have been possible. And if they had not got access to Franklin's information in the way that they had, if in fact Wilkins had respected Franklin's right to protect her data, it probably would have been Franklin's model that would have been the first published, because Watson and Crick would not have had access to such a nice image as the one Franklin produced, because Franklin produced an image that no one else was able to produce. But because of the way they got that information, Franklin was so ticked off with the way she was being treated that shortly after the papers were published, she actually left King's College and went to work for the British Coal Board. And unfortunately, a few years later, passed away from ovarian cancer. Almost certainly because she spent so much time in a room bathed in X-rays, and she wore no protection. She was known to walk through the X-ray beam on a regular basis, and she basically ignored the fact that it was known that X-rays were potentially carcinogenic, and it caught up with her as a consequence. It's very unfortunate because Franklin was a brilliant, brilliant scientist and probably would have made huge contributions if she had lived to continue her work, and particularly if she'd continued her work with DNA, she would have made some major contributions. But it is what is. So that's how the idea that DNA replication is semi-conservative was arrived at. And it was an important discovery because it was one of the two legs that supported Watson and Crick's model of the double helix. And so demonstrating that in fact it was semi-conservative was an important part of confirming Watson and Crick's model. So what we want to do for the rest of this section is talk about the actual mechanics of semi-conservative replication. Now, we're not going to dig very deeply into those mechanics. You may find that a statement that you're not necessarily going to agree with, but there is so much more that you could learn about the nature of the mechanics of semi-conservative replication if you were willing to take the time and make the effort. But we're not going to dig terribly deeply into those mechanics. But I do expect you to know the basic mechanics of how semi-conservative replication takes place on both the leading strand and the lagging strand in a replication fork. Also knowing what are the proteins and enzymes that contribute to the copying on a replication fork, and what the implications of the anti-parallel nature of the two strands is for the copying process. Because all of those are basic concepts that you need to be aware of for understanding DNA replication. So let's go ahead and take a look at what we know about those. So the first thing to realize is that the copying process is extremely rapid and very, very accurate. If we consider the DNA to simply be a string of letters, which basically is how we represent it, each nucleotide being represented by a letter. Imagine you copying out a sequence of 1 billion letters and making one mistake in the process. Because that's in essence what takes place in DNA replication. On average, the copying process itself makes one error in every 1 billion nucleotides. Considering that your DNA consists of somewhere between 3 and 5 billion nucleotides, the whole copying of your genome results in 3 to 5 errors on average. Now, there are other ways for errors to be introduced into the DNA. We'll talk about ways of repairing those a little bit later in this presentation, and we'll talk about the consequences of those errors when we do chapter 17. One of the reasons why bacteria can grow so quickly is because they incorporate nucleotides at the rate of about 500 nucleotides per second. So they are doing this very, very quickly. Eukaryotes do it a lot slower, about 50 nucleotides per second, but that's still a high rate. Again, 50 letters per second, no errors. Right? unless you are really, really good, you're not going to meet that standard at 50 letters per second, let alone 500 letters per second. But we know that's the rate at which it takes place. So the first thing to realize is that replication doesn't begin just anywhere, randomly on the DNA molecule. There are specific locations called origins of replication where the two complementary strands are separated or disengaged from each other by breaking the hydrogen bonds between the nucleotides in this region. Special proteins that used to be called single-stranded DNA binding proteins, but are now just referred to as single-stranded binding proteins, lock on to the separated strands to stop them from spontaneously linking up together again, which they could very well do, because remember, hydrogen bonds form spontaneously. They're just electrical attractions. So having these proteins to block them from coming back together is important. That then allows for the initiation of the replication process within what is now known as a replication bubble. And the bubble then begins to start to open up as a consequence of the action of some specialized enzymes that we'll talk about in a few moments that expand the replication bubble. And as that bubble expands, the copying of the DNA proceeds in both directions away from the origin of replication. In the case of bacteria, it moves in both directions, moving around the circular DNA molecule until the expanding bubble meets up on the opposite side of the DNA, resulting in the release of two daughter circular DNA molecules. And you can see the process taking place here, not on a bacterial genomic DNA, but on one of the smaller circular DNA molecules known as plasmids that are replicated at the same time using the same process. So here you can see the replication bubble. Here are the replication forks where the replication process is taking place, moving as the bubble expands in both directions around this little circular DNA molecule. In eukaryotic DNA, which is laid out in linear DNA molecules, the chromosomes, there are multiple origins of replication scattered down the length of the chromosomes. Some of the smaller chromosomes might have half a dozen or more origins of replication. Some of the larger chromosomes that we know of, certainly some of our larger chromosomes, may have upwards of 100 origins of replication. This allows the DNA replication to take place in a relatively rapid fashion because all of these origins of replication activate at the same time, forming replication bubbles that then migrate away from the origins towards each other. When they all link up, ultimately you have the two daughter DNA molecules, which become the two sister chromatids. Just in case you didn't make that connection, important connection to make. These two daughter DNA molecules become the two sister chromatids that are visible when the chromosomes condense and become visible during the beginning of cell division. And here you can see replication bubbles in a linear DNA molecule, and there are three replication bubbles here correlating to origins of replication. What this also illustrates is that those origins of replication are not necessarily located equidistant from each other down the linear chromosomes. They're just located in various locations down the chromosome. So this is the basis for DNA replication. It's what goes on initially at the origin of replication and then ultimately within the replication fork on each side of the replication bubble that is of interest to us from this point onwards. So the first thing to realize is what goes on at the origin of replication. So we begin to see some of the specialized enzymes that are involved in the opening up of the origin of replication and then the unwinding and unzipping of the DNA. So helicase unwinds and unzips the double helix. Now, because it's got to unwind it at such a rapid rate to keep up with the replication process, remember, DNA is being replicated at 500 nucleotides per second in bacteria, there is the potential for the DNA to overwind as it's being unwound. And if it does that, it's going to twist up into a knot because of the tension that's been created by this overwinding. So you've got a second enzyme called topoisomerase. So topoisomerase works ahead of the replication fork, and as it senses that the DNA is starting to potentially overwind, it simply nicks one of the two strands, which you can see here. It's created a cut in one of the two strands. As soon as you cut it, that relieves the tension. Then it immediately seals the cut up again so that the DNA is continuous to allow for replication again. So that's the role of topoisomerase. Primase creates I think I'm going to shift this down so that I can use this. Creates a short double-stranded sequence, which is the primer, using RNA nucleotides. And the reason for creating the primer is because of the nature of the DNA polymerase, which we'll get into in a moment. And then these single-stranded binding proteins, I've already told you a little bit about these. But these stop the unzipped strands let me just erase that. From rebinding, which they will do because of the fact that they're just held together by hydrogen bonds. So that's what all of these are doing. These are the major players. This is what is going on at the origin of replication. So helicase is what binds at the origin of replication, unwinds and starts unzipping the DNA. It starts moving along the DNA, and you can see it's moving along the strand here, moving from the 5' to 3' direction. That is not the direction in which the DNA is going to be copied, however. Because of the nature of the way the DNA polymerases operate, DNA polymerases always read the DNA in a 3' to 5' direction, but build the complementary strand in a 5' to 3' direction, which is why the primer is located on this strand. And you can see that the primer has a 5' end and a 3' end. The primase is simply moving in this direction, adding in the RNA nucleotides according to what the nucleotides are in the template strand that it's copying. I'm going to dig a little bit more into this in a little bit more detail, because we need to have an understanding of what the DNA polymerase is all about. So there are multiple DNA polymerases. In bacteria, there are 5 different DNA polymerases that are known. In eukaryotes, there are at least 3. Most of what I'm about to tell you relates to prokaryotic DNA replication, but the basic process is essentially the same in eukaryotic DNA. It just may be different DNA polymerases carrying out different tasks, but the basic process remains the same. So here you can see our template strand, and I believe that's how it's labeled up here. Let me move this box. Yep, so it says template strand up there. So this is the template strand. So this is the parental DNA that's being read. You can see here that we have a 3' end with the hydroxyl group. This is the end to which the new nucleotide is going to be attached. Which nucleotide depends on the nucleotide in the parental strand. So the DNA polymerase brings in the nucleotide and attempts to add it in by taking off these 2 phosphate groups and using this phosphate then to bridge to the 3' carbon of that sugar. That adds in the new nucleotide, but then before it's satisfied that it's done its job, it proofreads to make sure that it's the correct nucleotide, because DNA polymerase has a proofreading function to make sure that the right nucleotide is in the right location. This is one of the reasons why DNA copying is so accurate, because the polymerase not only inserts nucleotides according to the template sequence in the base pairing rules, but proofreads after the nucleotide has been added to confirm that the correct nucleotide has been positioned. And if an error has been made, it can remove the incorrect nucleotide and replace it with the correct nucleotide. But it can only add nucleotides to a 3' end of the complementary strand that it's creating. It is reading the template 3' to 5'. Now, you notice I've said here, template DNA is always read 3' to 5'. When we talk about transcription in chapter 17, which hopefully you've already started to take a look at, you'll see that in transcription, the template DNA is also read 3' to 5'. This is a hard and fast rule. Template DNA is always read 3' to 5'. The complementary strand, whether it be during DNA replication or whether it be during transcription, the complementary strand is always built in the 5' to 3' direction, because the polymerases can only add nucleotides to a 3' end. That's all they know how to do. So that's going to have consequences because of the anti-parallel nature of the 2 strands. Remember, I said that that would have a consequence when we came to looking at DNA replication. Now that pigeon, I guess, comes, or that chicken comes home to roost, because we do have to take into account the anti-parallel nature of the 2 strands as part of the copying process. So when we take a look at what is going on at the origin of replication, we find that on what is now known as the leading strand, a single primer is applied at the origin of replication by the primase. Then in bacteria, DNA polymerase 3, otherwise known as Pol 3, attaches to the template DNA, and using the template strand starts adding in DNA nucleotides to the 3' end of the complementary strand, starting with that primer as its complementary strand. And it continues to read the leading strand in the 3' to 5' end, so long as helicase keeps opening up the replication fork. So as far as the leading strand is concerned. So for the leading strand, is primed once, and continuously copied. By pole 3. So the question then becomes, if we're copying the leading strand, the strand that's being read 3' to 5', as a continuous copying process, what happens on the other strand? Because what we know from simple observation is that both strands at the replication fork are copied at exactly the same time, at essentially the same rate. So while the other strand is known as the lagging strand, it shouldn't be considered that that implies that it's copied at a slower rate, because it's not. It's copied at essentially the same rate as the leading strand. But remember, its orientation is anti-parallel to the leading strand. So even though the replication fork is opening up in this direction, we need to copy it so the replication fork is opening up that way, but this strand needs to be copied in this direction, leading away from the replication fork, leading towards the original origin of replication. So how is this done? It's done in chunks or fragments known as Okazaki fragments. So there's the name there. They're named for the Japanese postdoctoral student who worked in a research lab, again associated with the NIH in Washington, DC, who was responsible for doing most of the experimental work that led to the discovery of this copying process. When the paper was published, there were multiple authors, but because Okazaki was the individual who did most of the work, his name was the first name in the list of authors. And when a paper with multiple authors is referenced, it is referenced as Okazaki et al. So the only name that is ever mentioned is Okazaki when that paper is referenced, unless you're providing the full reference. And so these fragments became known as the fragments of Okazaki et al. And ultimately it was shortened to just Okazaki fragments. And because no one has come up with an alternate name for these are still known as Okazaki fragments, even though we try not to name things after people these days. Unless someone comes up with an alternative name that everyone is willing to accept, these will be known as Okazaki fragments for who knows how long. So what is the nature of the use of these Okazaki fragments? Well, remember, we need to be able to replicate the lagging strand going in the opposite direction to the replication fork. So as the replication fork expands, room becomes available for primase to come in and lay down a primer, which it does. And it lays down the primer, which creates a short strand of double-stranded nucleotides with a 3' end. Pol 3 knows what to do with that. So pol 3 comes in and attaches to the template strand and starts copying, reading the template in 3' to 5', and building the complementary strand 5' to 3', and it keeps going until, in this case, you move the text box out of the way here, until it comes up against the primer that was created either at the origin of replication, which would be this primer. And the one thing that pol 3 can't do is deal with those RNA nucleotides. Yes, it can proofread the DNA nucleotides, but when it encounters those RNA nucleotides, it doesn't know what to do. So when it comes up against a primer, which it will always do, it simply stops and detaches. Now, that would be bad news if that primer was left there. But fortunately, the primer is not going to be left there, because one of the other DNA polymerases, DNA polymerase 1, otherwise known as pol1, does recognize that those RNA nucleotides do not belong there. So it comes in and attaches, removes the RNA nucleotides and replaces them with DNA nucleotides. So at least now we have DNA nucleotides. But the one thing that it can't do is link up the gap between what is now a 3' end, because you're going to have a 3' there, and you're going to have a 5' there, and you're going to have a gap sitting in here. And the DNA polymerases simply don't know how to link those together. None of them know how to link those together. It's simply beyond what they're capable of doing. Fortunately, we don't have to leave the gaps there, because that would be bad news, because there is another enzyme called DNA ligase that does know how to seal those gaps. So once DNA pol 1 has finished replacing the RNA nucleotides, it disengages. DNA ligase comes in and links the 3' end to the 5' end between those fragments to create a continuous section of DNA. So when it comes to the lagging strand, it is primed multiple times. So the lagging strand is primed multiple times. And copied in fragments that are joined together. So the lagging strand copying is a little bit more complicated than the leading strand copying, only because of the anti-parallel nature of the two DNA strands. That's what leads to this. So we have to remember that, yes, the anti-parallel nature is a necessity because of the cross-linking between the nitrogenous bases, but that anti-parallel nature of the two strands now imposes the necessity to copy the lagging strand in a different fashion to the leading strand, simply because the copying can only take place with the template being read 3' to 5' and the complementary strand being extended in the 5' to 3' direction. So it adds an extra level of complication to the copying process, but nothing that you can't handle once you understand the nature of the copying process itself. So if we take a look at what goes on at a replication fork, when we consider the leading strand, It is primed once at the origin of replication. That creates the necessary double-stranded nucleotide section that the DNA polymerase requires with a 3' end that it can add nucleotides to, which it does using the template strand as its guide. Remember, it reads the template strand 3' to 5' and builds the complementary strand 5' to 3'. That's the leading strand, and it is primed once and then copied continuously. For the lagging strand, it has to be primed or reprimed multiple times. And it's reprimed as the replication fork expands. So as room presents itself, the primase comes in, adds in the primer so that Pol3 can copy the next fragment. Once it's done that and runs up against the previous primer, it gets out of the way, Pol1 comes in, replaces the primer, it then gets out of the way so that ligase can come in and link the fragments together. And this is going on repeatedly on the lagging strand. Of course, we have helicase still unwinding and unzipping, and down in this direction down here, we've got topoisomerase. Is somewhere ahead of the replication fork, removing the tension that's created by overwinding as the helicase continues to unwind the DNA. So these are all the players that are involved in DNA replication at a replication fork, and it gives you the basic ideas of what they do. And remember, this is all based on the bacterial model, specifically E. coli, if you must know, because E. coli is the lab rat bacterium. It's the one that we use predominantly for figuring things out because it's easy to grow. This is based on the E. coli model. Again, the names of the polymerases that are involved in eukaryotes may be a little bit different, but the basic process remains exactly the same, irrespective of whether you're talking about eukaryotes or you're talking about bacterial prokaryotic DNA replication. Now, one of the more interesting things that we've discovered about this is that this replication process is carried out by a replication machine, a complex of about 20 proteins working together, usually anchored to the inside of the nuclear envelope, through which the DNA is fed, rather than the enzymes sliding down the DNA and the enzymes being mobile and the DNA being static. It is the replication machine that is stationary or static, and it's the DNA that gets fed through. So this diagram illustrates what the machine looks like. If you take a look at the resource page, you'll find a video there, a BioFlix video, that shows you this complex in action as an animation. And you'll also find a link to a HHMI video, a Howard Hughes Medical Institute video, from which the BioFlix video was made. In fact, it's a simplified version of the HHMI video. But the HHMI video, it actually shows you pretty close to real time just how quickly these things are happening. It's very, very quick, and I believe I haven't actually sat down to count how many nucleotides per second it's adding because it's going too quick. But I believe it's actually was actually created to replicate in real time the rate at which nucleotides are added in a eukaryotic situation. Remember, about 50 nucleotides per second. It is very, very quick in that animation, but it shows you the leading strand and the lagging strand being replicated, and it shows you how the DNA is constantly being looped through the machinery, particularly for the lagging strand, to make sure that it gets copied correctly. They're very cool videos, and I would strongly recommend that you take a look at the videos. in the resource page. Because it's much more interesting to look at the videos than to look at this static diagram. It's really cool to look at. So I mentioned that there is the potential for proofreading, that the DNA polymerases can actually proofread the DNA to make sure that the correct nucleotides are being inserted. There's also a repair mechanism that takes care of structural errors in the DNA, which would represent potential sources of mutation that would occur outside of the copying process. The mutation would actually be propagated during the copying process because of these structural errors, but the structural errors themselves occur outside of the copying process. So one of the most common errors is what is known as a thymine dimer. So let me just make sure the pen is up. So the thymine dimer is typically formed by the action of ultraviolet radiation. So this is a very, very common structural change that can occur in the DNA in your skin cells, and therefore potentially lead to skin cancer if it remains uncorrected. But there are a family of enzymes called nucleases that patrol the DNA looking for such errors. Once they find the errors, they cut out the damaged strand. They don't cut the whole DNA, they don't cut both strands, because they need the undamaged strand to stay in place to act as a template. So they cut out the damaged strand, which in this case removes the dimer. DNA polymerase, probably DNA polymerase 1 in the bacterial situation, but it could be one of the other DNA polymerases outside of 3 and 1, which are involved in the actual replication process. Then comes in and replaces the missing nucleotides. But of course, that still leaves the gap between the 3' end of the repaired DNA and the 5' end of the rest of the strand. So ligase then comes in and seals the gap to finish the repair function. So in the end, you wind up with a restored sequence of nucleotides absent the original error that occurred. And these repair mechanisms are constantly patrolling the DNA to make sure that any structural changes that occur do not get propagated as a mutation. But of course, it's dependent on the nucleases, number one, finding the error, and number two, there being enough time to repair the error before copying takes place. So this is the reason why constantly exposing yourself to potential mutagens is probably not a smart thing to do, because you're basically challenging the repair mechanisms to fix any errors that might occur before the copying process takes place. So there are natural sources of mutation. I mean, we have radiation naturally occurring here on Earth. Some of the atoms in our body are radioactive and can potentially damage the DNA. But these repair mechanisms are active enough to, by and large, deal with all of that. It's when we challenge them by overexposing the DNA to potential mutagens and challenging them to repair the damage that's done that leads to an increased chance in the probability of damage being propagated as a mutation that could have serious consequences down the road. Then there's one other issue that we need to deal with, and that is that because our DNA molecules are linear, there are origins of replication at each end of the chromosome. And part of the problem with that is that when the primer is laid down on the lagging strand at the end of the chromosome, that primer can be removed, but it can't be replicated. That missing region can't be copied because there is no double-stranded nucleotides down here to provide a starting point for the DNA polymerase to fill in the primer. The primer can be removed because it can be recognized as an error, but you can't replace the missing nucleotides, which means that consequently, every time the DNA is copied, the daughter chromosomes get shorter and shorter, which means that at some point, if nothing changes, the chromosome area or the region of the chromosome that contains genetic information is going to get impacted and is going to start to get deleted as the chromosomes get shorter and shorter. Now, there is a system in place to compensate for this. It's called the telomere telomerase system. At each end of the chromosome, there is a repeating sequence of nucleotides called the telomere. So a telomere consists of 6 nucleotides. And there can be between approximately 75 and about 160 repeating copies. These are filled in by enzymes called telomerases during embryonic development because there's such a huge amount of cell division that takes place during embryological development that if the telomeres were not replaced, you would very quickly shorten the chromosomes to the point where the chromosomes would be damaged, and that would automatically trigger apoptosis, which we've talked about before, programmed cell death. Right? So during embryological development, the telomerases continue to replace the telomeres so that they don't, so the chromosome length does not get shortened. But somewhere during the first 6 months after birth, the telomerases in the vast majority of cells in the body simply stop working, which means the length of the telomeres in the vast majority of our somatic cells are fixed. The one place where that is not true is in the germ cells that give rise to gametes, particularly in the testicles, because men are going to make sperm once they hit puberty for the rest of their lives. And that means that the germ cells are undergoing multiple rounds of cell division, and that could potentially create problems with the chromosomes as a man gets older. Now, we do know that there are issues with men fathering children as they get older. We didn't think that there was an issue for a long time, but we now recognize that for older men, there can be potential problems if they father children at the genetic level, at the chromosome level. Another place where the telomerases are active is in the skin cells that give rise to the skin, because we are constantly replacing our skin every 4 weeks in fragments. So there's constant cell division going on in there. which is, again, one of the reasons why skin cancers are particularly bad, because these are cells that already have a mechanism for getting around apoptosis. Their telomeres are constantly being repaired. And you find this also in the cells in the bone marrow, where you are constantly generating the cells of red blood cells. That's where cell division needs to occur on an ongoing basis, so the telomeres need to be maintained in more or less good length. These are sacrificial pieces of DNA. They are there to be sacrificed every time a cell divides. But what you might imagine now, as you think this through, is that somatic cells have a limit in the number of times that they can divide. And that is true, they do. For most people, this is not an issue. But of course, if you force your cells to divide to repair cellular damage, that could limit the lifespan of the cells. And there is some indication that the length of the telomeres are also involved in aging. The exact correlation between length of telomeres and age is still very much poorly understood. There have been some people that have tried to create a direct correlation between lengths of telomeres and chronological age. But that is considered to be invalid, because we don't know enough about the actual nature of the role of telomeres in aging. What we do know is that for individual cells where the telomeres have been completely lost and the chromosomes, the coding regions of the chromosomes are now at risk, those cells will undergo apoptosis. We also know that one of the very common mutations that occurs in cancer cells is that the telomerase activity is restored. It's one of the reasons why cancer cells can grow and divide without confronting apoptosis. It is associated with tumor suppressor genes. Now, we do know how to switch on the telomerase system again. And in some little eukaryotic worms, that system has been played with and their lifespan has been expanded by a value of 3 to 5 times their chronological lifespan. And our telomerase system uses exactly the same mechanisms. So potentially there is a way of increasing human lifespan by turning on the telomerase activity. But remember, this is a double-edged sword, because we know that cancer cells also have reactivated telomerases. So turning on the telomerase system in somatic cells may simply make us more prone to cancer than we already are. So while this is something that we understand how to manipulate, it's possibly something that we don't want to mess with, simply because it's also associated with cancer, and it would simply increase the risk of cancer by turning on the telomerase system again. Because that is one of the most common mutations that occurs in cancer cells. It's why cancer cells are essentially immortal, because they turn on the telomerase system, which allows them to sidestep the whole apoptosis system and allows them to continue to grow and divide without being limited by apoptosis. So some food for thought there, I guess. But this is, again, another one of those systems that seems to represent a limit on human lifespan. And this, by the way, is a genetically inherited trait. The length of your telomerase regions on the ends of chromosomes is in fact an inherited trait. So if you have a family history of longevity, it's quite possible that your family has an inheritable long telomere region, or at least that has something to do with your longevity. We don't know exactly what the connection is between the length of the telomere region and longevity is. But we do know that loss of the telomere regions is correlated with aging. It is part of the aging process. Whatever that connection is, it remains to be seen. So that's the replication story. It's time for another break because we've been going for quite some time on that one as well. So let's take a look at a couple of concept check questions. I'll break, and when we come back from the break, I'll give you the answers to the concept check questions. So identify the two major functions of DNA pol 3 in DNA replication. That's the one that does most of the replicating process. If DNA pol 1 in a cell was non-functional, what would the effect be? What effect would that have on the synthesis of the leading strand? Now, we talked about it in terms of its role in the lagging strand. It's the one that replaces the primer. Would it have a role in the leading strand? And if so, what? And what effect may a non-functional pol 1 have on that? Let's take a break. I'll see you on the other side with the answers.